welcome to the microscope dissection for Bio 160 for Mr. Kaminsky's class. This is a much messier dissection than our frog and our fetal pig, so you're going to want to make sure that you have goggles instead of glasses, and of course all of your other required personal protective equipment. The first thing you're going to want to do is identify the sex of your microscope. So you're going to know the sex of your microscope based on the type of battery that is in it. If it is a Duracell battery, then it's going to be male, and if it is an Energizer battery, of course it is female. If there is two different kinds of batteries, then you have a hermaphrodite microscope. So what you're going to want to do is keep it supine and make an incision into the lateral side of the microscope, and you're going to want to just kind of make sure you get it pretty deep. And this is actually a hermaphrodite microscope. As you can see, we have two different batteries in it, so this one has mismatched gametes. So what you're going to want to do next is take your tweezers here and remove the gametes like so from your hermaphrodite microscope. And you're just going to want to set them to the side because you'll want to tell your teacher that you can identify them later. And right below, right after the gametes are removed, you will find the digestive sac of your microscope. This is a very healthy digestive sac of your microscope. Of course, a small one compared to other microscope digestive sacs you might see. This is the Microscopus geographia, the smallest type of microscope, the smallest species that you will find. It is native to North America, and it is what most American public schools use in their classrooms due to its small nature and ease of transport. As you can see, I've set the digestive sac over with the gametes so that your teacher can see that you can identify them both. So now we're going to cut into the base cavity of the microscope, and you're going to want to cut just along these lines here, like so, with your scalpel. Careful not to damage any of the organs on the inside, but also you want to make sure you cut through all of the muscle tissue. You may have to break a couple of the bones to get a clear look, and then you're just going to be able to just pop it off. And you're going to need to cut these to get a clear look. These are the veins. They're a little bit difficult, but as you can see, they can just cut. And you're going to want to take your scissors again, make a cut here, just to remove it in order to see better what you are looking at. Your teacher isn't going to ask you to be able to identify these, but if you care to know, this here is a vein, and this is an artery, and this is where they connect over here. So as you can see, we have here the duodenum of the microscope, and you're going to want to set that aside for your teacher. So this one is a bit hard to grab, kind of slippery, but if you can get a grip on it and grab it, this is the spinal cord of your microscope. Right there. And right next to your spinal cord, you can have to pop it out a little. You have your liver, which is the largest organ in the microscope. And I'll set that to the side here. Okay, and directly diagonal to your spinal cord, this is a really easy one to find. You have the brain of your microscope. So here we have, for your teachers, you have your gametes here, and you want to be able to identify your spinal cord, your liver, your duodenum, your brain, and your digestive sac. This has been your Bio 160 microscope dissection for Mr. Kaminsky's class. Join us next week for our tutorial on Bunsen burner husbandry.